Hello, everybody, and welcome back to the third installment of our Heritage Orchard Conference webinar series for this year. Today we have a great presentation by Cameron Peace coming up, and I'm going to hand it over to John Bunker, who was the one of our panelists in our first discussion as our moderator for Cameron's piece today. So you're on, John.
Okay, well, thank you, Kyle, and welcome everybody. I'm John Bunker, today's moderator. When I first heard about DNA testing of apples, I had a feeling of part fear, part dread, part panic, and part resignation. I had been learning to ID apples the old-fashioned way for a few decades, and I felt as though I'd made some progress. Now I was being replaced by a machine, or even worse, I was being replaced by an obscure chemical process that I could barely understand. Not only that, I was sure that this new thing would expose every error that I'd ever made. Over time, however, I came to the conclusion that actually I had to learn something about this process. I needed to understand this new competitor. Maybe, just maybe, there was something good about it. So I learned a little bit. DNA extractions and their sophisticated computer programs do it all by comparison. They actually don't know anything, but they're really good at comparisons. If you give them 2 or 3 small leaves of a northern spy you're uncertain about, and they have a northern spy on record in their database, bang, you've got a match. It's incredible. But what if they don't have a northern spy on record in their database? Then we have nothing with which to compare. There is no match, or as they say, unique unknown. And even if they do have a spy, some person at some point in time has to verify that that first benchmark really is a northern spy. That first ID can't be done by a computer. It has to be done by a person. And what about all those unique unknowns? They too need people to track down their lost identities. Not only that, even some of the largest and most prestigious apple collections in the world have errors. Who's going to clean up those mistakes? That's where we old-fashioned historian ideas kick in. It's up to us to declare that that first northern spy, Harrison, Colorado orange, Fletcher sweet, is the real deal. I was beginning to feel a bit more important. This was not a competition after all. This was going to be a collaboration. The Apple historian identifier and the Apple DNA scientist identifier. Over time, I began to think of all the incredible ways that we could work together. This could open all sorts of doors. In the summer of 2018, I wanted to put together a meeting of Apple historians and collectors in Geneva. Thomas Chow, the curator of apples at that time, and I began to make a plan. It turned out, Thomas told me, that there was going to be another Apple meeting at about the same time, this one led by last month's speaker, Dr. Gail Volk. Maybe we could stack them up together and some of the attendees from the first meeting would stick around for ours. Soon, Gail joined Thomas and me, and a plan was hatched. We met in September 2018 for a day and a half. There were about 20 of us. About half were academics, and the other half were historians, writers, cider makers, and apple collectors. Each of us made a short presentation of our current work to the group. I think everyone there found the meeting to be interesting, informative, and on top of that, quite fun. One of the participants was Dr. Cameron Peace, today's speaker. I had never met Cameron, nor had I heard him speak. I was enthralled. That led to a collaboration between Cameron and the Maine Heritage Orchard. In 2019 and 2020, we sent dozens of leaf samples to Washington State University for DNA SNP array testing. He also came to Maine Apple Camp in Maine in 2019. We have already learned much useful new information about our collection. In the next few years, we intend to test everything we've got and are planning more collaborations with Cameron and his team. Dr. Cameron Peace is fascinated by fruit, genetics, history, and what breeding offers future generations. He is a professor in the Department of Horticulture at Washington State University, and his research program aims to bring the benefits of natural diversity and genomics and the genomics age to fruit breeding. He uses new genome-wide DNA profiling techniques to understand the inherited attributes and ancestry of fruit trees. His current passion is reconstructing the family tree of all apple varieties, and the same for cherry. In Pullman, he oversees the student-run Palouse Wild Cider Apple Breeding Program. Cameron will speak for about half an hour, maybe a little longer. Then we'll take a break for a few of your questions, followed by the second half of his talk. At the end, we will have more time for your questions. Please write them in the question box on your screen. I'm delighted to present to you Cameron Peace.
Thank you, John. Thank you very much. Well, you've made a lot of the main points that I wanted to make, so I think we're good here. Thank you very much for the introduction and for being our moderator. Thank you too to Kyle for your amazing organizing prowess and all the things you do behind the scenes. And also thanks to the rest of the organizing committee for giving me the chance to talk today about apple identification with DNA, identity and parentage revelations using new tools. Pedigree, beyond this, pedigree and ancestry, another couple of major things that we get into, but for this talk, I'm going to focus just on the identity and parentage aspects. So thumbs up, you can see my screen, right? Yeah, great. Okay, mystery number one. In the suburbia of Pullman here. This is in my backyard, just back over here, up the hill. There are some really old trees there. They look like the remnants of an old orchard. They produce fruit most years. The one on the left here is an early season yellow fruiting tree. The one on the right comes out with late season fruit, which is striped. Both trees are probably grafted. They look pretty grafted to me, and they look like they're in a line as well. There's some other ones nearby, and I estimate they're at least 50 years old. And talking with other professors in the department years ago when I first came here, they said, oh yeah, there used to be old orchards up around the hills here in Pullman. So I'm impressed that they're still here, hidden in the backyards. And I want to know what they are. You know, what are these trees here? Obviously, they're different cultivars there. Each of these two. So what are their identities? Mystery number two. This is also in my backyard, just not far from the other ones, but a bit closer to the house. And this one is a late season striped, sweet fruited tree. It's probably not grafted, so I think it's a seedling, and I estimate it's around 30 to 40 years old. So what is its parentage? If it's a seedling, then I'm not expecting it to find the identity of it, but I'd like to know what its parents are. Okay, so there's a couple of mysteries that we'll come back to. So identity, why does it matter in the first place? Well, there's two major reasons. So one is genetics. The other one is going to be history. So the first one, genetics, inherited attributes, in other words, of what things have. So these are locked in. For some traits, genetics locks it in. So appearance, you know, the stripes and how much redness, or if it doesn't have any overcolor at all in the fruit. The acidity, resistance to scab and fire blight, lots of other things are really locked in for thanks to the genetics. For many other traits, it gives a likelihood, it gives a leaning, right? So how large the fruit's going to be, the yield, sweetness, some disease tolerances, those sorts of things. So it's very important, and very much so, therefore, because of the influence on traits, genetics determines our preferences to grow these trees, which ones we want to be growing there, which ones we buy, and which ones we eat. And the other reason it matters so much is because of history. So there are those stories and experiences attached to each of the named cultivars. It's really interesting to find out where and who first discovered each of the cultivars. That's why it's enjoyable to be reading John's books, for example, and why and how each cultivar spread. And it also attaches the stories or the experiences of anyone who's ever grown any of these cultivars and enjoyed them. So all of that's attached to any given identity. Why does parentage matter? Well, it's the same two reasons again. So genetics, so there's the inherited attributes, but there's an expectation that comes with parentage. So when we know the parents of something, it provides us a really good guess at how a child will perform and how it will appear. So it gives us a good guess at what it got from one parent or the other parent or both, and what it's going to give to the next generation. So this is the basis of breeding, and how breeders make decisions about what to cross, what parents to combine together to get to the next generation. But also for those of us who are not breeders, then it gives us an idea of what's happening, what it got, where it's going. And secondly, parentage matters because of the history. So it gives us information beyond just a chance seedling. So a chance seedling or a pippin is, it sounds pretty romantic, but really it just means orphan, right? So it's not connected up to what's behind it. So knowing the parents of something connects the tree to the stories and the experiences of each of those parents. So it doubles up. It's not just the identity of that cultivar, but even further back, all that's come down through the parents and their parents, etc. So it connects up the tree that you're considering here to a larger narrative. Okay, so I'm going to have 4 parts to this talk. First, I'll give you a brief introduction to the DNA profiling technology that's used, these SNP arrays, as John was mentioning earlier. Then identity, I'll talk about. Then we'll have an intermission see how much time we've got and get some Q&A going there. Then I'll get on to the second major aspect of the talk about parentage, I should say, sorry, not pedigree. And fourth is the thing that you'll be wondering is how to get your own identity and parentage information on your own mystery trees. And then we'll have some time for questions and answers later on. So first, I'd like to acknowledge key people and resources that have been involved in the work that I'll be explaining here. So Sten van der Zander, my postdoc in my program here, crunching all the data, crunching all the numbers, helping to get everything translated from A's and B's, as you'll see. Nick Howard, who does the same thing, has been extremely excellent collaborator and heavily involved in so many discoveries that we've been making. Like-minded other researchers from Europe, Eric van der Weg and Charles-Laric Durel, who also are heavily involved over the decades in pedigree reconstruction with Apple, and Gail Volk, who talked last week, who is always willing to share her ideas and data, and has made so many amazing contributions in this area of conserving and understanding Apple genetic resources. The work's conducted at Washington State University, and so of course they pay my salary. And the tools that I'm talking about here and a lot of the analyses that were done were within the Rosebreed project, which finished as a 10-year project, which completed last year, funded by the USDA. The USDA Plant Genetic Resources Unit in Geneva, New York, that holds the world's largest apple collection, and Green Global that holds the information regarding those, is extremely valuable, and the base material resource that's used. Dan Bussey's book, Illustrated History of Apples in the United States and Canada, took him thousands of hours, right, to do. It's already saved me hundreds and hundreds of hours using these volumes. So thank you very much, Dan, for putting that together. A lot of pictures I'll be showing are from the US Department of Agriculture Pomological watercolor collections. That's great that that's now online. Wonderful pictures. John's book, Apples and the Art of Detection. I draw on that in the talk here, and really inspiring to read. The Pomiferous website that has lots of great information, fairly new one that's come up, but lots of great information those guys are compiling on cultivars. A lot of material we've gotten from the Grove Heritage Nursery in Tasmania in Australia. And John Bunker and Todd Little-Sebold, thank you guys for introducing me to the world of New England apples and all the great people there and the Maine Heritage Orchard for the material that we've been checking out. Dave Benscotter and EJ Brandt, Lost Apple hunters, who've introduced me to that whole world. So thanks, guys. I know you're on now. And then all the folks from my lab, including current folks, AJ and Ty, who are really on the front line here doing a whole lot of the work behind the scenes. Okay, so first up, the DNA profiling technology. Let me give a brief description of how this works. So it's based on DNA. DNA, of course, is the blueprint of life, which has got the coded instructions for how everything that we do, our cells behave individually and all together to make up a functioning body of any given organism. DNA is inherited from our parents, this is very important, and it's arranged on chromosomes. Okay, so we've got chromosomes here. Each of our members of each pair of chromosomes, one comes from the father, one comes from the mother, and apples have 17 of those. Humans have 23, apples have 17. So what we can now do is DNA profile all along the genome, all the way down each chromosome. And so we use this technology called SNPs. So this has been a real key advance, and we get information which is hundreds and hundreds of genetic points all along each chromosome. And for every individual one of those SNPs, it reveals the genetic differences that's between those two chromosome pairs from the mother and from the father, but also the two chromosome pairs with any other individual. So a lot of genetic variation information is what it reveals. And SNPs themselves are not wonderful things. They only are binary, A and B, but because there's so many of them on these arrays that we use in this DNA profiling, we get really strong informative power, and it's from that high density of them that we are assaying across the genome. So those assays are the SNP arrays. So this allows us to test thousands of these SNPs very efficiently, and also an important point is that it's repeatably. So when we run these, we're testing the same regions for any new individual that we run, any new cultivar or seedling tree. So each of the SNPs that are on the array, we, a bunch of fruit scientists, determined which ones those were. There are 3 that are available for apple. The Rosebreed project developed the 8K, which means 8,000 SNPs are on that one. And then the folks in Europe developed the subsequent ones. And the one in the middle, I've put in green there, the 20K, we use that one because it's the most efficient for the most cost effective, and we don't need so many for most of the purposes, especially the purposes of identity and parentage. Because the one on the right costs like 4 or 5 times more. So really useful. So what this means is it's like a magnifying glass. You come in and can have a look at the plant, and we typically use leaf tissue. And so we come in and have a look at the DNA of that. So it gives us a look in. It's like we're looking in, peering in at the DNA to understand what's in there for any individual we look at. And any leaf will have the same DNA of that particular variety. Okay, so the information is just binary. It's just A and B. Yes, DNA comes in the flavors of A, C, G, and T, but the SNPs themselves just boil down to A versus B. So we convert all the data just to that. And so what's shown here, the first lot of SNPs at the very top of chromosome 1. And so we've got these that'll just go, down the page forever or across the page, across the spreadsheet for the thousands that we're looking at. So I'm just showing here in blue just a little snippet of what we might get for a particular test sample. Okay, so we run it and we get this sort of thing. All right, then we compare it. As John was saying, a lot of comparisons here you'll see is we compare it to the reference panel. And if there was something that was identical to that particular test sample that we have, then it's going to be exactly the same for every one of those SNPs, for the information we got for every one of those SNPs. So where it's AA on this one, it's AA on the other side, etc., all the way down. In this case, this test sample, which is something I do have in my backyard, not one of the ones I was showing you before, is matching Golden Delicious. Okay, so here we have Golden Delicious. Great, we'd say, oh, that's wonderful. Now I know what this thing is. Obviously must have been unripe on the left here. So completely identical for the entire genome when our test sample is matching exactly and it's identical to something in the reference panel. Okay, if something is not identical, then we'll get a couple of these SNPs. Some of these SNPs will come up as identical, exactly the same, but in most cases, they won't be. So we get many differences throughout. So it's very easy to identify none to detect and decide that something is not the same in the reference panel. We'd only need half a dozen of these SNPs, and we've got thousands to be able to show that something is different. Okay, now what if something is a parent or a child to our test sample? Well, what happens is one or both letters will be the same, but there'll always be at least one of the letters will be the same. So in the top one, you see there's an A that's in common. And the next one is a B that's in common, etc. So we'll get that all the way through the whole genome. So this partial match will be there for the entire genome. So in this case, the cultivar in the reference panel, Grimes Golden, is coming up as a match, as at least a partial match throughout the genome. So it indicates it's a parent or a child. Well, turns out that Grimes Golden is one of the parents of Golden Delicious. And so this happens, we get this at least one letter matching all the way through, is because children must inherit one of those letters. It's A or the B or the A and the A. And we call those alleles. It must have inherited one of those from each of its parents. So if something is not a parent or not a child, then we'll get many cases of at least one of the letters being the same, but lots of other cases where it's not, they mismatch. So it'll be a BB on one side and an AA on the other side. So we get a lot of these all across the genome. So that will easily show if something is not a parent or a child of our test sample. So Harrelson is an example here. Okay, so you might be wondering, well, parent or child, which way around does it go? How can we tell if we have some unknown samples, you know? So we put that individual in it. Do we actually have the parent of Grimes Golden, or is it the child of Grimes Golden? So which one's the child when we're comparing some samples here? Well, we don't necessarily know, but there's some ways we can really ascertain this. So the first is by considering the age of each of the two trees or the cultivars that we're comparing here. And so just the history that's attached to those and the information attached to those will help us determine which one came first, because often it leads to impossibilities being around the other way. Another way that really locks it in from with the DNA is if we have the other parent of the child that's in the reference data set. So when you get the three combinations, the AA, BB, and AB, there's only one way that this actually works. And so every time that the two parents are contrasting, the child must always be AB, and the other two ways of flipping that around just don't work. And so that makes it easy. The bigger the data set, the reference set that we have, the better. There's software that's available to handle these sorts of things. I prefer working with spreadsheets in Excel. First of all, the data, or eventually the data starts to look like this in black and white, but I don't like that either. So I like to play with colors and all sorts of other tweaks. So here we have our test sample, for example, on the top there in blue, and then it's comparing to how many of those letters match with each of these individuals. And this is just a little snippet from alphabetically from a longer list of materials that we would be comparing it to in the reference panel. So in this case, you can see Golden Delicious matches both letters all the way along. That's why it's an identical match, and therefore our test sample is Golden Delicious. We look at Grimes Golden there, which is one of the parents of Golden Delicious, then you can see there's ones and twos. So it matches at least one letter all the way along. As I was saying earlier, there happens to be a couple of children of Golden Delicious that are in this little set here, and you'll see that those also match at least 1 or 2 letters all the way along. So for everything else that is not a parent or child or not identical, then there'll be these mismatches, right? And we make those come up in red, and so there'll be plenty of those all the way through the genome. So that's really it. It's pretty simple. So John can actually now do it, and John, I'll be sending you some of these data sets, spreadsheets later on today. Okay, so I've been referring to this reference panel. So what's this reference panel that we have? Well, we have lots of cultivars that have, lots of materials, especially cultivars that we've brought together from all over the world, and lots of collections. So the Rosebreed project had at least about 200 cultivars, parents and ancestors of modern cultivars involved, 300 from Tasmania and other miscellaneous ones that have come in, 1,500 from Europe. And Nick Howard added 700 more on, and he's been consolidating all of these data sets together. And so that gives us a data set of more than 2,000 unique cultivars. There's some redundancies which are helpful among the collections and what people have been running. But this is what we've got from more than 25 collections worldwide. There's more than 1,000 that are going to be coming soon as well. So Nick Howard is adding about another 250 on. Gail Volk is in leading a project that's, she mentioned this last month, that's adding about another 600 key individuals that we don't already have in the collection, in our data panel there of heritage cultivars, and some Malus Aversii from Kazakhstan, carefully chosen ones there. And then from things that people have been sending in to me, we've got about another 150 miscellaneous things on their way. So that's what we get to compare everything against. And we want to and are continuing to add to this to be more and more comprehensive. Okay, so identity. Let's get into using this information to describe how we use it for identity. So how can trees be identical in the first place? How can our test sample come up with being exactly the same as something from a collection? Well, of course, it's from clonal propagation, or the grafting, cuttings in some cases, that have been done over the centuries and to spread all this material. And a lot of you guys are involved in doing that. And apple trees being long-lived also helps a lot to keep things alive, keep things going. And so any one of the propagules that comes from clonal propagation retains the same genetic makeup as the original tree. So that makes it really fun and easy, therefore, with DNA. So what it means is we can be growing the same cultivars that our ancestors did, which really fascinated me as a kid, and certainly as a young scientist. And that's why I really was excited to get involved in all of this and devote my career to this. Okay, I'm going to give you some examples now, some of the material that has been, samples that have been submitted and how it turned out. So after our first Heritage Orchard conference last year, 2 samples were submitted by one of the attendees there. First tree, they said it's similar, so it's in their yard, it's similar to Gravenstein, they said, bit different though. The upper tree, they don't know what it is. So this lower tree and an upper tree they sent in. And we're interested to find out what they were going to be. Well, it turned out both were grafted trees for sure, and both were identical with known cultivars. So the lower tree ended up being Melrose, and the upper tree was Macintosh. So they weren't suspecting that for the Melrose, and they're excited by the Macintosh, and so that was a fun one to do. Another example. An orchard and cidery in Western Washington submitted 2 versions of Tremlets bitter. So one is Geneva Tremlets that some of you might have out there, which is a bitter sharp cultivar, which means it's different to the original one from England, which is meant to be bitter sweet. And so in the US, it's also called false Tremlets. And orange pip and fruit trees thinks it might be an old English cultivar, Skirm's Kernel. And they submitted another one called Tremlets bitter, knowing it was different to the first one. However, this second sample is also bitter sharp, and so that also indicates it probably isn't the original Tremlets bitter from England. So the results turned out that they are not identical to each other, fair enough. That makes sense from how the trees looked and the fruit. The first one ended up being unique, this Geneva tremlets. It's got a parent-child relationship with Cap of Liberty. And the other one ended up not being, well, not Tremlets bitter, that's for sure, but ended up matching with Porter's Perfection. It's actually identical. It is Porter's Perfection, which is also a bitter sharp cultivar and likes to have fused fruit. So there we go. That helps sort out that situation. Lots of other opportunities, especially in the cider, the expanding cider industry, to sort out the identities of material that are being, things that are being propagated and sent around. John was mentioning earlier that he and Laura Seeger submitted lots of samples last year, and so I'll give some examples of how some of those turned out. So I've taken some stuff out of the book as well to explain some of these that were sent in, but Benton Red and Cherryfield, also known as Collins, and Blackhouse Red were three of the ones that were submitted. And it turns out that all three of these are Salome, an old cultivar. And so, yeah, that wasn't suspected. And that's something interesting for those guys. There we go. We've got an identity for those. Another example, a couple of cultivars, Nutting Bumpus and New Brunswicker. They are said to look very similar, and you can see in the picture there on the left that they do look very similar to Duchess of Oldenburg. So it was suspected that they might be clones, or maybe they're seedlings of. Picture on the right there is me at last year's Maine Apple Camp with Nutting Bumpus there. And oh, there it is. So I get a little photo of that one. So it turns out, yes, they are identical to both of those, Nutting Bumpus and New Brunswick, identical to Duchess of Oldenburg, which was an old cultivar that was introduced, a Russian one introduced from Russia in 1835, along with three other key Russian cultivars, especially to provide winter hardiness. It's Duchess of Oldenburg, also known as Duchess or Oldenburg, is the same as Borowitzki or Borovinka that's going around, but it's not the same as Shalamoth. Cultivar looks different. It looks similar, but it's different genetically. Several more of the materials that were sent in from Maine Heritage Orchard ended up being identical with known cultivars. So in blue here, in the quotation marks, are the names that were provided, have been given to these. So John, mostly I think, has come up with these names, but each of these matched with something that's already out there. So Orland Russet and Frank Arworth, both of those were found to be golden russet. Gardner Yellow, this one, it is Spencer, whatever Spencer is, but this might not be the real Spencer. This is the one in Geneva, because that really doesn't look the same. But anyway, both parents have also been determined for this thing. So maybe it's Spencer Seedless or something. I'm not sure. Nick Howard provided me the name for that one. Etc. Briggs Auburn was Northwest Greening. Givens ended up being Hubbardston Nonsuch. Sweet Red and Pumpkin Sweet of Orland were Sweet Delicious, and Pound Sweet is the real Pumpkin Sweet. So all old cultivars from mostly from the 1800s they ended up being. And then lots of other stuff with the Maine Heritage Orchard. So some of them ended up matching. Some of them that were submitted that were given cultivar names matched what they should be. So that's great. Others added to our reference panel. So that's useful. These are real cultivars out there. And that's great. We didn't have those in the reference data set, and so now we do. And then lots of others that have been given these names ended up being unique. So also useful. We can add those on now. They're not matching yet as identical to anything in the as an actual cultivar in the reference panel. We also tested out the Grove collection in Tasmania, which is the largest heritage cultivar collection in Australia. It especially represents Tasmania, and Tasmania was the origin of most apple cultivars. Of the apple industry in Australia, and so it's like and also grew the most in the origin. So it's kind of like the New England and the Washington for Australia. 201 accessions out of those 500 were carefully chosen and DNA profiled. about a third of those ended up being a different cultivar to what the label said. 3 were rootstocks. That pops up every now and then. But 56 were renamed in Australia through the decades, or maybe just mislabeled somewhere there along the way. So some examples, Bashford is just Northern Spy, so renamed as Bashford in Australia. It's just Northern Spy, etc. So some interesting ones there that are useful for those folks. 26 of them matched the reference panel, so it had the same name or same thing as we knew from a synonym of that cultivar, but it also provided many improvements to the reference panel. So it's a really useful collection that's got a lot of material that has been lost in the rest of the world. So that was good. So 106 additions. Some of them are really exciting, which I won't be talking about, sorry, in this particular talk, but some really great pedigree connections have been made from using this collection, such as we now know the parents of Granny Smith. Okay, sometimes there's some issues come up. So let's call these borrowers, identity borrowers here. And I don't mean the people, I mean the actual material, the samples themselves. And so here's an example of 2 identities, 1 DNA profile. So in the Grove collection, there's a cultivar called Red Gem. It's also known as Red Granny Smith. We ran both of these, they were identical, but they also ended up being identical to Arkansas Beauty. So the first one was from the Grove collection, and then the Arkansas Beauty, that's from Steptoe Butte, and this is a lost apple discovery that Dave Benscotter made. So the Red Gem or Red Granny Smith was selected in 1935, popped up there, whereas the one from Steptoe Butte, the US one, is a lot older than that. So we can figure out what's gone on here. So quite likely that in the early 1900s, Arkansas Beauty was introduced to Australia, but the records were lost. And so then this thing popped up and they've renamed it. So we had lots of material, lots of examples of that sort of thing. So that was a really interesting one. So great that that Dave had found Arkansas Beauty and that we had that now in the reference panel. So that's the key thing there. Okay, so what if we don't have a named cultivar in the reference panel? What do we do? So some of the name samples, as I was mentioning before, that have been submitted end up being unique, like those 106 cultivars in the Grove collection. Another couple of examples are Reg Mallard and Sari Sinap, which E.J. Brandt discovered, some lost apple discoveries. Those weren't in the collection, and so we've got the name now attached to them, Reg Mallard and Sari Sinap. So these become additions, therefore, to the reference panel. Okay, so now we have them, and this is the name that goes along with it, which should beg the question, how do we know they really are that cultivar? In fact, how do we know anything in the panel is true? And so this is what John was getting at earlier. So I'm going to repeat some of the points that John made before here, but yeah, we really do need you guys out there, the experts, and someone to make the historical connections. So the thing is, all the identities that we have in the reference panel are really just a working hypothesis, right? So they represent that name until we ever find a better explanation. And really, that's just the way science works, right? We don't say this absolutely is, we don't prove anything. It's just our best understanding. For most of the things that we have in the reference panel, our confidence is very high. This is because they're in cultivar collections and they've been vetted for quite a while. For any material in the collections and elsewhere, we have the passport or the source records that go along with it. Where did that tree come from? Who passed it on? Anything else about it? What its parents should be, etc. The verification from experts really key. So the identity experts, growers, a lot of these things are commercial cultivars as well. And so if someone's growing that and selling it, that helps lock that in as well. And where the information we get matches historical records, and where the DNA profile also matches independent samples and also matches the parent records, then that really helps out in making sure that increasing our confidence a lot that we've got the right thing. So when we have enough evidence like this establishes the type profile for that individual. And we can then check others to see if they are true to that type, true to type, as it's said. So it's important that we record, anyone out there keeps recording and retaining the source information. Where did that tree come from, and everything we can about it. Where did you get it from, that grafted tree? And do as much ground truthing, and thanks for that term from Todd, as much ground truthing as possible is very, very important to do, to make sure that we have well-established identities in our reference panel. So what about seedlings? What's the identity of seedlings then? Well, seedlings, being ungrafted trees, give unique DNA profiles. And that's what John said earlier. So unless, of course, they've been propagated and sent around, and independently they pop up in the reference panel. But otherwise, something that you suspect to be a seedling that's out there will just about always come up with a unique DNA profile. So these are the types of things that you find along roadsides, railway lines, fences, on stone walls, along creeks, out in the wilderness, as long as it's not an old orchard, abandoned orchard. And then these things will be seedlings. Could be on the edge of an abandoned orchard. So when you get these, of course, give them names and propagate them, share them if they're great. So that's excellent. So let me give the identity of a celebrated seedling here. This is the old apple tree at Fort Vancouver in Washington. So this was the oldest known tree until it died earlier this year, and was 194 years old, which is pretty awesome. It's long been celebrated and propagated and shared, but it just was never commercially grown. So it didn't have a name, didn't come up in the pedigree of anything further, didn't have descendants, etc., come up in cultivars. So it just was a seedling, but around for a long, long time. So we ran this one, did the DNA for this one, and it came up, as you'd expect for a seedling, with a unique DNA profile. It wasn't something else. It wasn't a direct, grafting from England, for example, and an English cultivar. So it confirmed the historical records of that this thing did arise as a seedling. And it has been recently named. Now it's got a cultivar name, Fort Vancouver. So thanks to Paul Stash for connecting me up and providing the samples and getting me involved in working on that one. Okay, so now I'll resolve mystery 1. So this is the one where we've got these 2 grafted trees up in the backyard, the remnants of an old orchard here. So I ran these samples, and the early season yellow one came out as Lodi. Okay, so cultivar from commercially released around 1924. And the other one, this late season striped one, ends up being wine sap, which is what one of the old professors here suggested it might be when I said we've got an orchard. He said, well, I used to grow wine saps a lot in Pullman, so that's good. So mystery solved. Great. All right, intermission time. Time for some of your questions, or at least me to take a breather and coffee sip.
Okay, so here's a question. What is the error rate on a SNP target?
So, okay, I'll assume that this means out of the thousands of SNPs that we're looking at, how often is it wrong, an individual one wrong? If you mean something else by that, then maybe rewrite the question, but I'll answer that one anyway, because that's a good question. So it's very low. It's 0.1%, something like that, with the raw data, and that's just the data when we just accept it as it automatically gets scored and put that into our data set. So it's a very, very small number. Whereas a mismatch between a parent and a child will be, between 2 things that aren't related to each other directly, will be a lot more than that. Will be at an order of magnitude or 2 more than that. So it's very easy to tell the difference between something that is just a random error in the scoring that we have and a true different individual.
Okay, there's a couple questions about sports. One is, what does a sport look like when you do this? And the other question is, has anybody looked into Ben Davis, Gano, and Black Ben Davis and that controversy, which has been around for over 100 years? People have been wondering about those three apples.
Okay. What was the first one again? Sorry.
Just the how does a sport show up?
Yeah,
when
sports.
when
Yep,
you test
yep,
the
yep.
sport?
Okay, right. And so then the Ben Davis Gano thing is a good example of that.
Yeah.
So sports will have the same profile, the exact same DNA profile. So we can't distinguish the differences between those. That being said, it also means that it doesn't matter when a cultivar has a child, it doesn't matter which sport that child came from. It still always looks like just the base individual. So Red Delicious, for example, and Starking Delicious, and Red Chief, and Big Top, and Red Top, all these various sports of Red Delicious, for example, if they have children, they all just seem like, Hawkeye, or like the original Delicious. So it makes no difference there. Sports being mutants that have appeared in someone's orchard, typically on a branch, that then have been propagated on, that just had something that looks different, right? There's all mutations going on all the time, from one branch to another, and all these propagules, grafted trees that get spread around. And but if any of those little mutations lead to something that we can see, such as more redness on the skin, or makes it a week later or a week earlier in the season, then we will pass those on. But otherwise, they're not actually any different to any other branch that's been propagated, or any other sport. So the Ben Davis Gano thing, we have Gano here in our research orchard at Washington State here, and so we've just collected some samples just the other day to test that. But I assume it is just Ben Davis for now.
There's also an apple that's been kicking around called Black Ben Davis, and some people think that's the same too.
Yeah, so we'll check these out from experience with other situations like that. Lots of other cultivars have things like that. It ends up being the same thing. So if it seems to be the same in just about every case, it is the same. But they're not children, for example.
What about triploids and tetraploids? What can you tell about them?
Okay, got some examples sort of coming up in a bit, but triploids, they take a bit of extra effort, and Nick Howard's been getting into this, but we can tell the parents that have gone in to make them, and it'll be one of the parents hasn't split in half, and so it's passing on both of its letters, both of its alleles, and then the other parent halves as normal, and so it comes together to make 1.5 times the normal genome size of apple. So we can tell the parents of triploids. Triploids, we have zero evidence that triploids have led to any children. So there are no pedigree descendants from anything triploid. Okay, so I end up, because I'm a geneticist and following things through pedigrees all the time, when I see a triploid, it's a dead end and I lose interest in it, because I know it's not going to pass on to be connected up further down the generations. It could be a sibling and an uncle and a great aunt and all that sort of thing with things, but it won't be a parent or a grandparent or a great grandparent or anything.
Do you want to? So we're at 10 of the hour. Do you want another question or two, or should we?
Yeah, give us another question or two. Yep.
Okay, so there's a question about Golden Delicious and its possible self-pollination and how that figures into all this.
Okay,
and maybe you could just talk about self-pollination in general too. Does that exist in apples, and if so, how does that show up?
So I'm interested in, I've been wondering about that too myself over the years. I'd like to do a project on that, to check that out. So if anyone has anything that they feel really sure has come up as from a selfing, then get in touch with me. But we have zero evidence. Nothing has ever come up that showing that it only has one parent, that the mother and the father are the same thing in apple. There's zero evidence for now, up till now, from DNA profiling, including the DNA testing technique that Gail was talking about last month, that we can get viable cultivars for starters from selfing. So I suspect that most cases of apparent selfing are just the bees, being a bit cheeky and bringing in stuff from elsewhere. And there's a lot of there's a lot of paternity that's wrong as well. And so that is probably my main reason for saying that, is that the bees do a lot. Even when we purposely put pollen onto things, bees do all sorts of other things behind the scenes.
Okay. Now, let's see. Here's one more. Has any analysis been done on recently rediscovered historic American cider varieties, Provençal and Talifero?
Yeah, so
To that.
we have ad hoc bits and pieces. We've run some cider things. We tried to get a project up and going last year, didn't get funded yet, to really systematically go after that and figure everything else out in cider. We're putting that in as part of another project shortly this year and into next year. So we can do that. We have run a Talifero, for example, and a couple other things like that. And there are pedigree connections and all sorts of things coming up there. But if there are particular questions or big issues that anyone has about certain cultivars of cider, especially if they've got two things they want to determine what's going on, like the Tremlets bitter example I showed before, then please get in touch with me.
Okay, now here's a question about land races, and are unique samples ever determined to be a land race? Well, maybe the questioner is wondering about, is that is it turns out to be a specific species, or something? I don't know.
Yeah, so if we're talking about species, there aren't any cultivars that are derived from the American species. So Malus, Angustifolia, Coronaria, and Ioensis, and also Fusca on the west coast. So none of that has come into any modern cultivars at the moment. Breeders are, including in our program, are purposely making crosses along these lines, but it'll take a few generations to get something commercially viable, even for cider. So there's nothing there that's come in, if that's what the land race means. Otherwise, if it's the type of material that was just spread as seeds by colonists, that type of land race, then that's what all of this stuff is, or most of it. And I'll get into a little bit more of that in the second part of the talk here.
How about just a quick word on ornamental crabs and how do they figure into your work?
So again, just like the other cultivars, the other species in America, I mean, they only a little bit barely or a tiny bit get into the pedigrees of certain cultivars. So that there is a little bit of that. They make for some really fascinating stories, like Honeycrisp has some wild in it. We do work on this in a big way. It's not part of any of the talk I'll be giving now. We also have in the Palouse Wild Cider breeding program here, we cross with wilds like crazy, all sorts of crabapple species. So there's lots of valuable attributes that wild species have, and we encourage the use of those in breeding, and we can detect those quite easily as well. The bits of the DNA that come in from those, they stand out a lot compared to other normal cultivated apple.
Shall we?
That's its own talk, basically. I could have a whole talk just on that. So that's just a very quick answer.
Okay.
Yeah, let's move
continue on with part two.
Yep.
Okay.
Yep. So thanks for the questions, folks, and the key ones that we didn't answer yet. Hopefully we'll get to those afterwards.
Yeah, we'll have more time, and I'm keeping track of the questions. Keep sending them in.
Great. Okay, so part 3, parentage. Okay, so we talked about identity. Now let's get onto parentage. So most of what I've been talking about here is about American cultivars, historic American cultivars. There was a bunch of slides I ended up cutting out about Tasmanian stuff, so stick it on the American ones. But there's great paper that we talked about Gail Volk talked about last month. We put the link up last month, at least anyway. It's Historic American Apple Cultivars, Identification and Availability. Wonderful paper. It's freely available. So if you just do a Google search for that title, you'll find it and get access to it. So the information here in the next slide comes from, the key points I'm making come from this paper. But some of the cultivars that we have in the US were brought from Europe directly as grafted trees, but most originated as seedlings that were first planted in the US. So some of the earliest ones that originated in the US are High Top Sweet, Roxbury Russet, and Rhode Island Greening. So these are back in the 1600s, which is pretty amazing. It's probably why they look like they have so much dust on them there. So early colonists got seeds and they planted these widely. That's why most cultivars end up coming from this seedling material. So this was highly encouraged to help quickly establish homesteads. Sometimes, of course, some of these ones came up that were quite desirable, really interesting one way or another. And so these were detected by the homesteaders and others, clonally propagated and named and shared. So seeds, therefore, were the basis of these seedlings. So where did these come from? Well, they had to have come from available fruit that are in the hands of somebody there, someone in the US here. So where did they come from? Well, sometimes from Europe. And so the old apple tree, the Fort Vancouver one, is an example of that, of where the fruit was actually in England and someone had put the seeds into their pocket and come to the US with those seeds. But usually and increasingly they were from local trees. So the cultivars that were grown, these grafted ones originally planted that were brought from Europe, but then grew up and had their own fruit, their own seeds. Okay, so these were seeds then from which cultivars are we talking about? It can't be just from anything. Right? It's got to be things that historically make sense, etc. So what about these really old ones that were brought over from Europe? Golden Russet, Golden Harvey, Gillyflower, French Reignette, these sorts of things. Any of these? Okay, perhaps. What about some of those first cultivars from the US that were originated in the US? High Top Sweet, Roxbury Russet, etc. Any of those? Well, I'll tell you first of all, not those first three, High Top Sweet, Roxbury Russet, or Rhode Island Greening, because all three of those are triploids. So unfortunately, because of that, we don't have any descendants from those. And then there's, more recent ones, but still they're a couple hundred years old in most cases here. Jonathan Winesap, Ben Davis, were these sorts of things, the parents. So hopefully you're thinking about that. Where did these seeds actually come from? Hopefully you're also thinking, what's the father as well? So the seeds come from, you get it out of an apple of the mother, but to get a seed, you need two parents. And so each seedling's a unique combination of those parents. The father, of course, is the pollen that is typically brought by the bees. So don't forget the father when you're thinking about parentage and what's behind something. Too often I read stuff and it's only the mother. It's all people often think about. Which way round, which was the father and which was the mother? It doesn't matter genetically. So the attributes still come out the same. How that tree performs, there's no evidence that it matters which way around it goes. It does matter though for checking the reliability, matching it up with written records. And that's because the mother, when it's recorded, is usually correct. So that's where someone got that fruit from that tree. Usually they know that or they remember that and recorded that. So usually that is correct. Father though, often we don't know because the bees that brought the pollen. And even when it's suspected or whatever, often that's wrong as well. So the DNA is figuring out what a lot of these things really are, the other half of the parents there. It's really easy to determine parentage with the SNP array, as I was showing you earlier. Each seedling has that unique combination of its two parents and the DNA is coming together. So they come out with these unique DNA profiles. And the children also are matching each of the parents across the entire genome, as I mentioned earlier. And it's really easy to identify parents, as long as the parents are in that reference panel. And another useful thing is if we can identify one or both of the parents, it can help really date and historically place the seedling. That makes it really interesting as well. In some cases, it can turn out that, you know, it's not a seedling. It might actually figure out that it's an actual cultivar based on those particular parents. So yeah, quite easy to figure this stuff out with the SNP array. Okay, so I'll give you an example here, first of all, and go through several examples. So here's one from Steptoe Butte in Washington. So Dave Benscotter, after the last Heritage Orchard Conference last year, he led a post-conference tour out to Steptoe Butte. So a bunch of us went along to that, and he showed us this really interesting tree. I think, Dave, I hope this is the one that's out there. It was that one that we're talking about here. Anyway, he suspects that this is Hoover. Okay, so I went, well, I know someone who could do some DNA profiling of this, so I grabbed some leaves and took them with me. Turns out that it's not identical to Hoover, at least the Hoover that's in the Grove collection in Tasmania. And it didn't match up with anything else, at least yet, out there. However, the parentage did come out, was detected with these really cool parents, Blue Pear Maine and Wolf River. That's pretty awesome parentage, I reckon. And so that's good. But here's a bit more info, or a bit more of my suspicion is, and I'll talk with you, Dave, more about this, but I think that actually your tree is the true Hoover. I think that the Grove one might be wrong. And it's based on parentage connection that this individual ends up having with some other things, the individual from Grove. So I think it actually is Hoover. Anyway, that was really useful to lock in the parentage there of this thing. Another example, Wealthy. So this tree was raised in the 1850s. The records say, if you jump on the internet and look up old historical records, you'll see that it says that it was suspected to be seed from Siberian crab, which is thought to be where its winter hardiness came from. Very important in Minnesota, of course, and the northern parts of the US and Canada for growing apples. So very important cultivar, this one. It's in the pedigree of lots of other things. It's an ancestor of lots of other cultivars that are around these days, especially in the really key Minnesota breeding program. Well, it turns out nothing to do with Siberian crab. Duchess of Oldenburg, Prost, Jonathan is what led directly to Wealthy. So the winter hardiness quite likely therefore comes from Duchess of Oldenburg. So that's been an interesting one. Here's another one that we now know the two parents of as well, Delicious. So this was raised in the 1880s. The records say that it sprung up from a rootstock when the Scion cultivar died. It's been the most produced cultivar in the US since the mid-1950s until just last year. Finally, it's now #2 and going down. And of course, there's many sports that have been developed from Delicious that are more blushed than the original. And they have, as I was mentioning earlier, they have the same DNA profile. So when we get the DNA out of the leaves and check it, it just comes out with the exact same thing, whatever the sport is. Okay, so what are its parents? Wine sap and white winter pear Maine. So both of these are old American cultivars, and so it's got a very clear American lineage pedigree here. Delicious does. So next time you come across a wine sap, now I've got some terrible examples up in the backyard here, or white winter pear Maine, and if you somehow manage to get them together, check it out, taste them, and look closely and go, oh, red Delicious popped out of this thing, out of these two. In some cases, we can't identify one or both of the parents. In those cases, what's the missing parent? Well, we then suspect that it could be a unique seedling tree that was out there. Maybe the bees brought some pollen from something that's long since dead, just a one-off, right? But the parents of that thing, we might be able to figure out. That's a different talk. Or it's a cultivar that is out there, but we just don't have it yet in the reference panel. And so more and more as we add to the reference panel, we fill in these missing individuals, these missing parents. So Golden Delicious is one that we have determined one of the parents for. Grimes Golden, as I was mentioning earlier, we don't know the other parent yet. So this has been a very popular cultivar for a century. It's a parent of so many, direct parent of so many key cultivars that are out there in the supermarkets these days. And therefore it was chosen to have its whole genome sequenced, which was published in 2010, which at the time was only like the 4th plant genome in the world to have been sequenced. So Grimes Golden, parent of Golden Delicious. Another one, Jonathan. So this is a top 10 cultivar produced in the US. It's been around a long, long time, almost 200 years. I suspect that it's had the longest run of being in the top 10 of any cultivar. And one of its parents is Asopus Spitzenberg. So again, next time you have those, look at them next to each other. I've done that, and you can really see the differences between them. So let's look at Asopus Spitzenberg now. That's another key one. So this was recorded as an accidental seedling in Albany, New York, not far from Asopus, and it's said to be Thomas Jefferson's favorite. We know one of its parents, French Rainette. This is the only American cultivar that we've found yet that connects directly with this very, very key and old cultivar, French Rainette, which has been around for more than 500 years and is in the pedigree of so many things. Really key cultivar, this French Rainette. So Asopus Spitzenberg is probably the oldest, perhaps, landrace in the US. And that picture, that watercolor, looks beautiful. Here's a picture from one of our local trees that looks a bit more realistic. But still, yeah, a decent apple. Tastes good. Mind you, they did paint those watercolors with warts and all, but that was a lovely painting, a lovely looking apple, that one. Okay, some examples here from the Maine Heritage Orchard. So we found one or both parents for some of these cultivars. So here's a photo I found online from you guys, from the photo that was taken at the Common Ground Country Fair there of several cultivars. Sorry, several individuals that were found. You know, what are these? A couple on here we ran, samples were sent in, and we ran these. Cranberry Island ended up being Duchess of Oldenburg crossed with Zuzov. So a couple of old Russian cultivars there. Talked about Duchess before, but Zuzov is another one from Russia that was brought in a bit later. And Green Monster is another one, ends up being a child of Tolman Sweet. Then some others, some more. So the Sweet Red and Pumpkin Sweet of Orland, which I said was Sweet Delicious, that ends up being derived from Deacon Jones and Delicious, a couple of late 1800s cultivars. This Gardner Yellow is Delicious crossed with Macintosh. The Salome one, the Benton Red, Cherryfield, Collins, Blackhouse Red, those ones, they are Westfield. Westfield is one of the parents of all of that, Salome and those individuals, which are all identical. So that's a couple 100-year-old cultivar, Westfield. Westfield, seek no further, is another name for it. Then a couple of russets. So Wyndham Russet ends up being a child of Asopus Spitzenberg. At least one of the parents there. And then Hunt Russet, which is an actual cultivar that's been around for quite a while. One of its parents is another really old one, similar to French Rainette from France. It's been around a long, long time. And then we have its other parent is an imputed one, this unknown founder 13, as Nick Howard calls it. And I'll come back to that a little bit later. But it's got some pretty prestigious parentage there, Hunt Russet. Some key cultivars out there, American ones, are still orphans. We haven't determined their parents. In some cases, we've determined at least one of their grandparents, so we're reconstructing their pedigrees, but for the purposes of parentage, we're still looking for those parents, so we're always eager to add new, really old trees to our reference panel so we can figure out ultimately what the parents are of these things. So as a reminder, you know, the parents could be something. We might never find the actual parent itself, one or both parents, because they could have just been unique seedling trees long since gone. But hopefully there'll be something that we add that we later find. So that includes, yeah, Northern Spy, Ben Davis, Macintosh, Winesap, etc. Some cultivars we find have many, many children. So Golden Delicious, I was pointing out, is a parent of lots of modern cultivars that are in the supermarket. So this has been a key founder. So many things radiate directly off it. And then a lot of cultivars have then descended from that too. So that's a good example. A couple of other ones are Delicious and Cox's Orange Pippin. They also have been majorly used by breeders, basically in the last 100 years or a little bit more, and have just passed on some really useful genetics, at least for the modern industry. So these are all direct children, and there's so many more. There's more than 50 key cultivars in the world that are directly descended from each of these. So these are kind of more modern, or at least well-known individuals, but there's, going back further in the pedigree, there's some other situations where we have key founders. So we've found some ancient ones that have been discovered or imputed. So French Rainette, I was pointing out before, we consider this one as the grandmother of everything. So it's the direct parent of many really old cultivars, ones that have been around 300, 400 years. So French Rainette got passed around a lot. It must have got passed around a lot or brought to the US, or at least its seeds brought over, because it is also the parent, like of the Serpa Spitzenberg, but then the grandparent and great-grandparent and great-grandparent, great-great-grandparent of so many American cultivars as well. Really, really, really key. The keyest founder, the most critical founder of world apple production for the last hundreds and hundreds of years in the pedigree, including American. Then there are a couple of these situations of these imputed founders that are the direct parent of lots of things. So this unknown founder 13 that I said was one of the parents of Hunt Russet, it's the parent of lots of other russets as well. And then we have this other one that Nick calls unknown founder 6, is the parent of so many cider cultivars as well. And so this is from the work of Nick Howard to detect these. So it's really fascinating. We're hoping we'll find these. I mean, the fact that so many things came from it means that these will be actual cultivars. This won't be a one-off unknown tree that's now dead. It should have been propagated and spread around. So we're quite hopeful we will find these particular ones. And you can tell from the numbers that they go up. There's a bunch of other key unknown founders out there. Unknown founder number 1, which Nick had called number 1, was actually found. It was in the Grove collection in Tasmania. And that had 29 really old cultivars came off it. So I've been talking a bit about pedigrees. I'm talking about parentage, you know, as you go further and further back in generations, and you might be getting a picture of, like the way I see it, is like this, that everything really, I think, connects up into one big family tree. And I'm hopeful that all of this can be reconstructed, although it's pretty much a lot more like this. It's even more complicated. There's so many more things out there, but I don't see cultivars as independent. I think everything all comes back in and connects all in. And so that's what we're working on, is sticking it all together. And the idea is to be able to figure out how any given tree, any cultivar, any accession in a germplasm collection, or any backyard tree or whatever it is, that we can figure out using DNA profiling where it fits in the family tree. So that's a talk for another day. And all that stuff for apple that we've been doing, we do it for cherry too. So lots of amazing stuff is coming out in cherry. Okay, so now I'll resolve mystery number 2. This was the suspected seedling tree, probably not grafted, chance seedling that's come up in my backyard. way back, and it ends up being the inauspicious parentage of Golden Delicious crossed with Delicious. So it's okay. It's not that good, this particular tree, the fruit. So I'm not going to name it. I'm not going to propagate it. So don't ask for it. There are lots of things out there that are Golden Delicious and delicious, but it was good to know. Useful to find out. So what about your seedling tree out there? What's the parentage of that? Well, send your samples in, right? Let's find out. So that gets me onto the last part, how to get the identity and parentage information on your mystery tree. So here are the steps. Let's go through them very quickly. Is you get the leaves, put it into a bag or a tube if you've been sent one of those, and you put it in the mail and send it to me. And then you wait a bunch of time, and then you get emailed your report, and I'll tell you what the identity is or the parentage, at least. And then you send a check. So it's $120 currently, used to be $100. Sorry, it's gone up. And thanks to service providers out there changing from labs that we send stuff to. But anyway, $120 per sample. Our steps is we receive your tissue, just a brief thing just so you know what goes on the inside here. We get your samples, we then transfer those into these plates. When they get filled up, we send those off to a company who then does the DNA extraction. We used to do that in our lab, but now we're sending that off. We wait patiently there. Once those all come in, then we process those a little bit further, put them into a different plate and send those off to another company. And then we wait and they run the array and get all that information. All the A's and B's come back. We get back the raw data. We convert all that lovely raw data into the A's and B's connected with each of those SNPs, and then play around in Excel to figure out the identity and parentage. Just wanted to give you an overview of the aggregate results so far from all the samples that have been sent in the last couple of years that we've been looking at. So about half of them are already in the reference panel. About half of the ones that have a name on them that have been sent in, named cultivars, believed to be cultivars, they end up being new additions. So those have been quite useful. And then anything that has been, it's like, it doesn't match with any known cultivar, or it's known, suspected to be a seedling. Most of those do come up as being unique. And so those are also useful new additions to the reference panel. So that's been really useful to get those and add to everything, but gives you an idea of how often we'll be able to determine the parentage and figure out the identity of material that gets sent in, that you send in. So if you are interested in getting this done at $120 per sample, then you can request the sample collection instructions from me. Please email me and I'll send that to you. And give me an idea of how many samples you're thinking of. And I always want to hear about your stories, by the way, and see the photos of the trees and the fruit and stuff like that. That's why I do it. This is not anonymous to me. I'm not a lab to switch off and just treat things like numbers and letters and stuff like this. I care about all of your stories as well and these trees. So I'm fascinated to hear about it. So I want to help solve your mysteries. Coming next year, hopefully, we're developing a cheaper alternative for doing this identity and parentage. So it should only be about a quarter of the price. This is efficient for especially when you want to do lots of trees. Let's say, everything in the old orchard or everything along the creek, for example, especially if you can get it even cheaper. Especially if you suspect that the trees are just common cultivars or that they're just seedlings from common cultivars, then that's going to be especially useful for this new quicker technique and cheaper technique. And if it ends up being something really fascinating, we can go on to the full DNA profile for it. It's not available yet, though. So don't hold your breath, but we're working on it to really open it up to more and more people. And with that, I will take questions.
Okay, well, thank you. I think one of the most important questions that came in was the question about a central database and what is happening with this information? I know that Gail Volk is working on some things. Can you tell us about that?
Yes, I can. So I anticipated this question, and I have a couple of slides. So yeah, I totally agree. There's a need for a single repository that we have reliable cultivar information for all the various people that care and want to know. There's all the stakeholders out there. So, you know, who has and what are the descriptions of the true to type specimens? Where are they? What do they look like? What are their, you don't need to know the DNA profiles, but at least know that it's attached to it. You know, what are the parents and the pedigrees there? Make sure that's correct, and then we connect it up with that. Because yeah, a lot of what I read on the internet is not correct. What are the genetic predispositions, right? So what does the DNA tell us about what the fruit quality will be or the appearance, etc. Consolidating all the various performance records out there. You know, just saying something as crisp is not really useful. Let's get that together and harmonize that, because that's been collected under all sorts of circumstances, and have a network as well as part of the database of who has what, right? What are the ongoing trials? Who are the experts out there for particular trees or groups of cultivars or whatever, or regions? What's the commercial availability of each of these cultivars? And this came up a couple of months ago, I think in the first session, someone put in the chat, a national sharing network of science, you know, that sort of thing. I like that phrasing. That's really good. So we're working on a research proposal to do this. I'm leading this to actually develop it for apple, also for cherry. And three main things here that I think are important to incorporate in here. So identity and parentage diagnostics, being able to do that, those more efficient tools to do that. Pedigree reconstruction, so we can connect everything all up and integrate that with historical ground truthing. And the cultivar database itself, so the interface with everybody that wants to use it and get the information, and so have that single repository of that reliable cultivar information. So yeah, absolutely, heavily working on that, and that's the big research thing that I'm going after. We have a meeting later on this week, and John's going to be on that, and a few other people, Gail Volk, to discuss what can be done in the meantime over the next year before any new funding kicks in as well. Because with the genome database for Rosaceae, which is here at WSU, Dory Maine runs that, we have some opportunities to not wait around for new funding, but we can move ahead in pulling all this together. So yes, absolutely think it's very important to do this.
A couple of questions about specific varieties. One is Hawaii, which was supposedly a cross between two triploids, and also Ribston Pippin, the supposed parent of Cox's orange pippin. Can you tell us anything about either of those? Yeah,
if Hawaii is in our database, I think Geneva ends up being one of its parents. If the person asking that can email me, I can look it up more specifically about that one. Ribston Pippin, a good example of incorrect information out there, or record that's not that someone just made it up, but it's the ideas that have been out there for a couple of centuries. Is certainly not the parent of Cox's orange pippin. It's got a whole bunch of genome that it shares with Cox, but it's not a direct parent-child situation. And so Ribston Pippin, being a triploid, is not a parent of anything, as I was saying before, as well. So it's a pedigree dead end. Cox, yeah, Margil is a really old cultivar that hopefully some of you have, because it's also really, really delicious, just like Cox. That is one of the known parents of Cox. We don't know the other one yet, but we're reconstructing some of the pedigree behind it. But yeah. That's where Cox gets its awesome flavor and other things like that.
Okay. So here's a question from Dave Benscotter. The Lost Apple Project would like to be put in touch with leaf donors who send in unique apples. Is that possible? So I guess the general question would be, how do we connect with one another when somebody sends in something that for me might be a lost New England apple or the Pacific Northwest or down in Colorado or somewhere.
Yeah, so, right, so coordinating it regionally would be a really great idea, right? I think that's what Dave's offering, for example, for this neck of the woods over here. So if, people contact the, the local expert, the regional expert, first of all, and as they are, I know with Dave already, people contact him all the time about what they've found and what it might be, that can help, the expert then can help determine whether that's something that could be worth looking into further DNA wise, or else, not, that it's more of something that it's more likely just, there's an easier way to determine what it is. Like, oh no, that's just Macintosh, or that's just wine sap or something like that, by looking at the fruit, for example. So that would be, that would be really useful if we could coordinate that somehow. So yes, contacting Dave is, if a lot of the stuff goes through Dave before it comes to me, that sounds awesome to me. As we have been doing. So thanks,
okay.
Dave.
Can you, we're starting to run out of time here. There's a question about, what if the tree is dead? Is there anything you can do?
So if it's dead, totally dead, as in not just, last month, but dead for many years, then I don't think so. There's some amazing things that have been done in getting DNA out of all sorts of stuff, other organisms and things that have been around dead for a long time. So it's probably possible. So I'd say, collect it and put it into a bag and, scrape off some trunk or something. If there's brown leaves, actually, you probably can get it out of brown leaves. We've been testing that out and that seems to be okay. But if it's more of just the skeleton of a trunk, maybe get in and scrape some off on the inside. At least have that, better than nothing. Maybe the technology will sort itself out at some point.
Well, I think we're running out of time here. There are a number of other great questions, but one I think that would be interesting to some people is another apple variety is Gravenstein, probably one of the most famous of all apples grown in the United States, in North America. What can you tell us about, and there are many sports and so forth. What can you tell us about Gravenstein?
So I think that's a triploid as well. I should have had my file open. I knew I was going to get these specific questions, but I think Gravenstein is a triploid, and so again, pedigree, dead end. It can have lots of sports, but will not. It's not in the. It's not a parent or grandparent, etc. of anything.
And while you mentioned the word sport, there was some confusion that some people asked about. Can you tell us again genetically what a sport is?
So it's not a parent or child or anything like that. It is just a detected mutant where one of the genes, sometimes a couple of the genes, on a branch, as that branch has been developing, have changed and has led to a detectable visual difference. So the rest of the genome is entirely the same. It's just at that one little spot that it's different.
Okay, now I think, how about as a last question or request, what haven't you told us that you'd like to tell us today, and what would you like all of us to be out there doing now?
What I haven't told you about is all the great stuff on cherry. That's just as much. I mean, there's fewer cultivars out there, but it's such an interesting, fascinating story as well, where again, the American cultivars are the basis of the world industry. So like Golden Delicious and Red Delicious, for example, are the basis of so many things out there in the world for commercial production. Similarly, in cherry, the American ones are the basis. And then we've looked at the pedigree back behind those and connecting all that up. So that's a really fascinating story too. And what I want you all to do is make crosses, right? Consider what you want for the next generation. Think of be Johnny Appleseed, right? Not just propagating up these cultivars that you know and love, but put the best of those together, right? What if you get Blue Pear Maine and Wolf River, right? And whatever else, Cox and Wine Sap and whatever, cross them and get to the next generation, grow up the kids, grow up a whole bunch of seedlings, and at least 10, let's say, of any given cross that you make. So at least the seeds from one fruit, and grow them out in your garden and pick the best. You'll be fascinated by what you find. So all of us can be breeders.
Well, thank you, Cameron, very much. This was great. And thank you, everybody, for joining us today. And we'll be back again in about a month. Kyle, do you want to add anything?
No, I was just going to recommend that everybody tunes in for our next webinar on November 11th. And it's going to be on apples and their uses, past and present. And that webinar is going to be a panel discussion moderated by me with Steve Miller from the University of Wyoming and Katrina Mendre from Montana State University. So it should be a good one.
Great. Thank you.
Thank you.
Thanks everyone for attending. If we didn't get to your questions, you can email them over to me and I will try to put you in touch with the best person on our Heritage Orchard Conference Committee to get these questions answered. Thanks for tuning in, and we'll see you all next month.
Thanks, John. Thanks, Kyle. Thanks, everyone, for tuning in.
Thank you. Thank you.
Bye.